Hello friends, I am MD Harun Al Rashid and this is my YouTube channel Harun Biozone. Today in this video, I will discuss 10 very important and tricky MCQ questions for CSIR, NET, SET, GATE, CUCAT, SSC, PSC, all over all the competitive exams based on biology. So let's go. The first question is, so which of these enzymes in glycolysis, so all the enzymes are used in glycolysis, do not take part in an irreversible reaction? Do not take part in irreversible reaction. That means all the enzyme take part in reversible reaction. So where these enzymes are required? First in glycolysis cycle, glucose is converted into glucose 6 phosphate by hexokinase. Clear? Then again reversible reaction to produce fructose 6 phosphate. Then again irreversible reaction phosphofructokinase phosphofructokinase fructokinase produce fructose on 6 bis phosphate. Okay, and this pyruvate kinase use at last use phosphoenol pyruvate to pyruvate. Here pyruvate kinase is used. The three enzyme hexokinase, phosphofructokinase and pyruvate kinase. And another glucokinase also used in here. The glucose to glucose 6 phosphate is a irreversible reaction, but here the hexokinase do not take part in reversible reaction. Phosphofructokinase also do not take part in reversible reaction. Phosphofructokinase also do not take part in reversible reaction. Why? Because the end product of this reaction, suppose glucose to glucose 6 phosphate, this glucose 6 phosphate inhibit hexokinase. So then do not reversible reaction occur. Fructose on 6 bis phosphate also inhibit phosphofructokinase, so do not reverse. Pyruvate inhibits the pyruvate kinase, so do not reverse reaction occur. But here one enzyme is glucokinase. Glucokinase only found in liver. Remember, only found in liver, but hexokinase found in all over the tissue, all over the body. बॉडी की हर जगह में हेक्सोकाइनेज मिलता है, मगर ओनली इन लीवर ग्लूकाइनेज इज फाउंड. And this glucokinase do not inhibited by this glucose 6 phosphate. Okay, this glucokinase do not inhibited by glucose 6 phosphate. So glucose to glucose 6 phosphate is produced, and also glucose 6 phosphate to glucose is produced by glucokinase. Clear? So the glucokinase do not take part in irreversible reaction. They show reversible reaction. The function of hexokinase and glucokinase are same, but hexokinase take part in non-reversible reaction, but glucokinase take part in both reversible reaction, and only found in liver tissue, but hexokinase found in all over the tissue. Why it take part in reversible reaction? Because glucose 6 phosphate do not inhibit glucokinase, but glucose 6 phosphate, high concentration of glucose 6 phosphate inhibit hexokinase. Clear? The correct answer is D. Glucokinase is the enzyme that do not take part in irreversible reaction but take part in reversible reaction. I think you understand. Next question: which of these following enzymes does not take part in pyruvate dehydrogenase complex? The first we understand what is pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex is made by three enzymes: E1, E2, enzyme 2 and enzyme 3. Enzyme 1 is pyruvate dehydrogenase. Pyruvate dehydrogenase. Dehydrogenase. Okay. And E2 is the dihydrolipoyl transacetylase. Dihydrolipoyl transacetylase. Transacetylase. And 3 is dihydrolipoyl dehydrogenase. Dehydrogenase. These three enzymes are combined to form pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex. So here, which of these following enzyme does not take part in pyruvate complex? So glyceraldehyde 3-phosphate dehydrogenase is a glycolytic enzyme that do not take part in the synthesis of pyruvate dehydrogenase complex synthesis. Okay. Next question. Name the inhibitor of glycolysis. So which inhibitor inhibit the glycolysis process? First, fluoroacetate. So what is the function of fluoroacetate? Fluoroacetate is a pesticide. Okay. That inhibit the enzyme that inhibit the enzyme aconitase. That means this enzyme, what this enzyme required? Acetyl in Krebs cycle, acetyl coenzyme A and oxaloacetic acid is combined to produce citrate, and the citrate against produce isocitrate. Clear? In the presence of aconitase enzyme. So this aconitase enzyme if this aconitase enzyme is inhibited by iodoacetate, the fluoroacetate, this enzyme fluoroacetate, this citrate do not convert into isocitrate, so inhibit respiration in Krebs cycle. Clear? First, so what is the function of iodoacetate? Iodoacetate is an inhibitor of glyceraldehyde 3-phosphate dehydrogenase. Iodoacetate inhibit 3-phosphoglyceraldehyde to produce 1,3-bis-phosphoglycerate. Okay, in glycolysis in the presence of phosphoglycerate dehydrogenase. Phosphoglycerol dehydrogenase enzyme. Clear? That convert NAD plus to NADH plus H plus. This iodoacetate blocks this enzyme, inhibit this enzyme by iodoacetate. So iodoacetate blocks the glycolysis. What is the function of malonate? Again, malonate in Krebs cycle, we know in Krebs cycle succinate convert into fumarate. by succinate dehydrogenase hydrogenase enzyme that convert FAD plus to F to FADH2. So this enzyme is inhibited by malonate. So malonate also inhibit Krebs cycle. What is the function of actinomycin? The actinomycin D or actinomycin that inhibit transcription inhibitor. It is called transcription inhibitor. By binding to DNA, and block RNA polymerase. Okay, so here the correct option is iodoacetate, because only iodoacetate that inhibit this reaction of glycolase 3-PGLD2 and 3-phosphoglycerate. But malonate also Krebs cycle inhibitor, and fluoroacetate also aconitase inhibitor, and actinomycin D is a transcription inhibitor. The correct option is B, that is iodoacetate. Next question: how many molecules of CO2 are produced by acetyl group of acetyl CoA in citric acid? So here pyruvate, two molecules of pyruvate convert into two molecules of acetyl CoA, coenzyme A. Clear? By dehydrogenase enzyme, decarboxylase enzyme, where CO2 is released, two molecules of CO2 released, NAD plus is converted into NADH plus H plus. Add what is the functional group, prosthetic group for this enzyme? Thymine pyrophosphate, FAD, CoA, coenzyme A, etc. So add. So here only two molecules of CO2 are produced during this conversion. Okay, but in all of our TCS cycle, 6 CO2 is produced. 6 CO2 produced. I think clear your concept. Next, what is respirosome? What is respirosome? What is respirosome? Respirosome is the functional unit of electron transport system. Respirosome is the supramolecular complex of complex 1, 3 and 4. Means complex 1 means NADH, dehydrogenase, complex 3 is cytochrome reductase, and cytochrome oxidase. Complex 2 is a free floating substance. The supramolecular complex means when the small subunit combine to produce a large subunit, that is called supramolecular complex. Okay, so these three complex are combined to produce the respirosome. Respirosome is functional unit of electron transport system. What is the function of this? What is the function of this respirosome? Means reduce oxidative damage. reduce oxidative damage of electron transport system or electron transport chain. Number 1. Number 2, increase metabolic efficiency. Increase metabolic efficiency. This is the two function of respiration. The correct option is the supramolecular complex 1, 3 and 4. The 2 complex is free floating. 2 is free floating complex. Floating complex. Okay. The next question is name of the physiological uncoupler. Name of the physiological uncoupler which stops ATP synthesis. So suppose this is the electron transport chain. Clear? This is a complex 1. This is complex 2. Complex 3. Complex 4 and complex 5. This is outer mitochondrial space, this is inner mitochondrial. So here NADH2 is converted into NAD plus 2 proton plus 2 electron. Here FADH2 is converted to FAD plus 2 proton plus 2 electron. These protons are transferred from matrix to intermembrane space. And normal, this proton also, they have ubiquinon is present. Ubiquinon. This proton also transport to intermembrane space, and these electron are transferred from complex 1 to complex 2. But here this electron also transfer. Then here plastocyanin is present, electron transfer. Then complex 4, complex 3. Okay. Then is ATP synthesis. The 3 proton is transferred through this ATP synthase and produce one molecule ADP converted into ATP. Clear? Here this is a normal process, but in the presence of uncoupler agent, this proton do not transfer. These protons do not transfer through this way, but that means this proton transfer in any other way or do not transfer, but not this ATP synthase, not this F0F1 complex. So it is called uncoupler. Okay, this couple are dissociate. So what is a 2,4-dinitrophenol? 2,4-dinitrophenol. 2,4-dinitrophenol is the first discover uncoupler agent. Coupler agent in 1930. This is uncoupler agent. Number 2 is dicomarol. Number 2 is dicomarol. So what is dicomarol? Dicomarol is a anticoagulant produced from plants, coagulant, and used in the treatment of vascular thrombosis. Or blood clotting thrombosis. So this is not uncoupler agent. FCCP. What is the full form of FCCP? FCCP. FCCP full form is carbonyl cyanide. Carbonyl cyanide. P-trifluoro. P-trifluoro. Methoxyphenyl hydrazone. Methoxyphenyl hydrazone. This is the full form of FCCP. FCCP is a lipophilic compound. Lipophilic cationic proton transfer. Lipophilic cationic proton proton transporter. What is its meaning? इसका माने क्या है? Here normally these protons, ये जो प्रोटॉन है वो जाता है कहाँ से है? F0F1 कॉम्प्लेक्स से मैट्रिक्स में जाता है, मगर दिस इज अ लिपोफिलिक, मतलब दिस मेम्ब्रेन इज फॉस्फोलिपिड मेड बाय फॉस्फोलिपिड बाइलेयर, so it is made by lipid. Okay, so in the presence of FCCP, the proton, normally these proton do not transfer in this lipid bilayer membrane, but in the presence of FCCP they can carry the proton through this lipid membrane bilayer. So it is called lipophilic cationic proton transporter. So the proton do not transfer through this ATP synthase but through the lipid bilayer membrane. So do not ATP is produced. So prevent the oxidative phosphorylation. Oxidative phosphorylation is prevented. And another is thermogenin. Thermogenin is also also called UCP1. UCP1. It is a 33 kilodalton protein present in inner mitochondrial membrane. This is a thermogen is a physiological uncoupler. It is present in inner mitochondrial membrane. These walls are synthetic, but this is the natural or physiological uncoupler agent, thermogenin, present in the inner mitochondrial membrane. I think you completely understand this question. So let's go the next question. What is the Curie cycle? Is a glyoxylate cycle? Is a gluconeogenesis, lactic acid cycle, or citric acid cycle? The Curie cycle is a lactic acid cycle. How? Suppose this is liver. Curie cycle means this is liver, and this is muscle. The normally glucose during respiration, glucose convert to pyruvate, and pyruvate again convert to lactate in anaerobic respiration. This lactate transfer to liver, and lactate is convert to pyruvate, then pyruvate to glucose. Okay, then again this glucose transfer to muscle. This cycle is Curie cycle. Because why Curie? Because Carl and Gerty Curie first discovered this cycle. So according to their name, this cycle is called Curie cycle. So it is also called lactic acid cycle. 8 number. Which of the following is correct for the Pasteur effect? So what is Pasteur effect? Utilization of glucose for fermentation. Or number 2, increase glucose consumption by yeast in anaerobic condition. Fermentation favor aerobic condition more than anaerobic. Increase consumption of glucose by yeast in anaerobic condition. The Pasteur effect means Louis Pasteur first observed that yeast consume glucose. Yeast consume glucose. Yeast consume glucose. Glucose aerobically less efficiently. Less efficiently. But anaerobically more efficiently. Clear? So this is called Pasteur effect. The correct option is increased glucose consumption by yeast in anaerobic condition. This is the right one answer. Next question. Mark the incorrect statement coenzyme Q. Coenzyme Q. Where is coenzyme Q is present? Coenzyme Q is a electron transporter. Okay, electron transporter. No, sorry, not electron transporter. It's a prosthetic group. Not prosthetic group. It shows ubiquitous in nature, right? It is also an ubiquinon. Means in electron transport chain, this is the NADH complex 1. Here the ubiquinon is present. The ubiquinon is a electron transporter. Ubiquinon. Okay. It is a ubiquinon. Q refers to the quinone chemical group. So here incorrect is D. It has a protein bound to prosthetic group. Not protein. It is not protein bound to prosthetic group. It is electron carrier. Electron carrier in respiration or electron transport chain. Next question is, out of the following, which is not the inhibitor of the electron transport chain? Which one is not inhibitor of electron transport chain? The first, what is rotenone? Rotenone inhibit complex 1. Inhibit complex 1. Complex 1, that is NADH Q reductase. So prevent electron transfer from iron sulfur complex of NADH Q reductase to ubiquinone. Clear? Number 2, it produce reactive oxygen complex. Oxygen complex that damage DNA. Clear? And mitochondria. The complete function I describe here. Mitochondria. Okay. Rotenone naturally found in jicama vine plant. Jicama vine plant from the family Fabaceae. So it is electron transport chain inhibitor. Number 2, actinomycin A. What is the function of actinomycin A? Actinomycin A inhibit complex 3 cytochrome reductase. Inhibit complex 3, that is cytochrome reductase. That bind to the QI ubiquinone I region of complex 3. Then number 3, cyanide. Cyanide. The cyanide bind to iron in cytochrome A3 of complex 4. Okay. Prevent cells from using oxygen, and that prevents cells from using oxygen O2 and producing OTP दोनों प्रोसेस को बंद कर देते हैं। तो क्या हुआ, अगर ऑक्सीजन एब्सेंस हो जाएगा तो फिर डेवलप एनारोबिक कंडीशन and in anaerobic condition produce lactic acid, so acidosis occur. Clear? And in lactic anaerobic condition there is no ATP is produced, so or very least number of ATP is produced, so the death of the human body occur immediately. Malonate is inhibit TCS cycle. Malonate inhibit TCS cycle by when succinate to fumarate is produced. Succinate to fumarate is produced. Fumarate. The enzyme requires succinate dehydrogenase that convert FADH2 to FAD plus. This enzyme is inhibited by malonate. So here the only enzyme malonate do not inhibit electron transfer chain. The correct answer is D. Okay, I think this is very helpful to you. Thank you for watching this video. Thank you.